This is a lecture on the genetic regulation of longevity in mice from September 18th, 2020. And today we're going to talk a little bit about some specific proteins and genes, particularly INS1, insulin receptors, as well as the growth hormone receptor genes. And we're going to talk about the relationship between insulin signaling and growth hormone signaling and longevity that's been studied in mice. And then a little bit about how the findings that we have discovered about genetic regulation of aging and longevity in mice can relate back to aging and longevity in humans. Okay. And so as of right now, in mice, 30 to 40 genes that affect longevity have been identified. And there is a table here from your book, Table 5.4, that includes some genes with mutations in mice that have been shown to extend lifespan. And I've highlighted some of the ones that we've talked a little bit about before. So you can see mTOR here. This mutation, or this hypomorph in mTOR, can actually extend lifespan in the same way that the mTOR pathway extends longevity in yeast. And then you'll notice here some components of the insulin signaling pathway have also been implicated in increasing longevity. But there are some additional genes that are going to discuss today, including the growth hormone receptor gene, as well as some genes that create dwarf mice, particularly the Snell dwarf, and how those smaller sized mice, those dwarf mice, and as well as the ones with that lack growth hormone receptor, have extended longevity as well. So we're first going to start by talking a little bit about those insulin genes and the insulin pathway and how it relates to longevity in mice. And so one gene that I want you to know from this particular lecture is the INS1 gene, and it encodes an insulin receptor substrate. It's basically a homolog to human insulin. And mice that are a total knockout from birth for insulin 1 or INS1 are actually unable to survive, but they have done conditional knockouts where they knock out insulin 1 after development and then see how that affects lifespan in mice. And in these mice, there is indeed an extended lifespan. But what's interesting is that they only see this extension of lifespan in female mice and not male mice. And they're not totally sure why there's this gender difference and why insulin 1 knockouts might have an extended lifespan in the female mice but not the males. And so just as a reminder, if you knock out or decrease the amount of insulin or insulin receptor substrate in flies and in worms, you will also extend lifespan. And so this is similar to what we see in some of the other genetic model organisms. And then another gene in the insulin pathway that has been studied in mice is the IR, the insulin receptor gene. So not the substrate or the ligand for that receptor, but the receptor itself, which just like DAF2 and the insulin receptor in flies, INR, the insulin receptor in mice is a transmembrane receptor that can detect insulin on the outside and transmit that signal to the intracellular environment. And what we've seen in worms and in flies is that if we have loss of function mutations in the insulin receptor, there is an extension of lifespan. So if DAF2 mutations as well as INR mutations extend both worm and fly lifespans respectively. But when we look in mice, whether they're females or males, loss of function mutations, like you can see in the survival curve here, don't actually affect the lifespan of mice at all. And that is because we're talking about insulin signaling in mammals, which is much more complex and intertwined with other pathways than it is in sort of the lower organism or the invertebrates. And so you can see here some of the effects that insulin has on protein metabolism. So the formation of proteins and then how that can actually affect growth. And it's just to illustrate that insulin has a lot more functions and it has its hands in a lot more things, in a mammal or a vertebrate like a mouse than it does in a fly or a worm. And that's one thing that also becomes unfortunate about finding all of these longevity and aging genes in invertebrates, is that sometimes the results don't necessarily correlate back to the vertebrates or mammals like mice, and therefore are not relevant to humans either. And so this mutant mouse actually has a loss of function mutation in the insulin receptors throughout the whole body. But there is some effect on lifespan if we take a specific type of insulin receptor only expressed on certain cells and then knock that out or decrease that receptor's function. And that's what we see in these mice known as FURCO mice. And FURCO stands for fat-specific insulin receptor knockout. And so these FURCO mice lack insulin receptors, but only in their fat cells or the adipose tissue. And so you can see here that compared to just that general loss of function insulin mutant, FURCO mice do actually extend lifespan. And so that mutation in insulin receptor on the previous slide in every single tissue didn't have an effect, but this fat-specific insulin receptor decreasing its expression or lacking it can actually extend lifespan compared to wild-type mice. And this is an image from a paper studying the physiological changes that happen in these FURCO mice that lack the fat-specific insulin receptor. And so the KOs here are those FURCO mice. So you can see male knockouts in black and female knockouts in this darker gray color. And what was interesting is that compared to wild type, both male and female mice, the knockouts that lack this fat-specific insulin receptor have more lean tissue and less fatty tissue. And so it's possible mechanistically that the decrease in adipose or fat storage that's seen in the FURCO knockout mice compared to wild types is actually what's causing this extension of lifespan. And that this increase in the lean tissue can contribute as well. Because as we know, there is a link, at least in that's seen both in mice as well as in humans, where increased fat can actually decrease lifespan. And so that is one potential mechanism for how longevity is conferred to the FURCO mice, but it hasn't been well tested yet. And the reason that this decreased fat storage exists or happens in these mice is that what insulin receptors will do is bind to insulin and then have it taken into cells at a higher rate. And once it's taken into cells, it can be converted into fats, especially when there's an excess of glucose, right? So insulin receptors will sense the glucose levels and cause glucose to be taken in from the blood. Glucose can then be converted into fat. And in mice that lack the insulin receptor, they can't sense this glucose, therefore they can't take it into the cells, and they can't make or store this fat as a result. And it's possible that, once again, that mechanism is helping confer the extension of lifespan seen in the FURCO mice. And to make things more complicated, when we talk about insulin signaling as well as insulin-like growth factors, or IGFs, and we talk about them in mammals, there's a lot more interplay between the different organs and what controls signaling or secretion of insulin and IGFs. It's not just one thing like it is in worms, where nutrients will signal the secretion of insulins in response to a positive kind of food signal. In mammals, what we have is nutrients can actually signal the pancreas to secrete insulin, but in addition, nutrients also will affect the brain and cause the pituitary gland to ultimately secrete growth hormone, and then growth hormone will stimulate liver to secrete insulin-like growth factors. And insulin-like growth factors will both be able to act on the cells to promote cell growth and proliferation. And so there's this interplay between direct sort of signaling of nutrients to the pancreas to make insulin and a more indirect or neuroendocrine pathway that involves both the brain as well as endocrine secretion of insulin-like growth factor. And what's important is that this connects the insulins to growth hormone signaling as well, which is a completely separate signaling pathway. But what it also means is that growth hormone is also likely implicated in longevity. And this has actually been shown in mice as well. And so there are some mice, the growth hormone receptor knockout mice, or the GHRKO mice, which you can see here on the far right, where they've knocked out the receptor for growth hormone, and therefore tissues aren't able to respond to that growth hormone and grow larger, and cells are not proliferating as effectively. These mice, these knockout mice, have a much smaller size, as you can see here on the far right, compared to wild type, which is the far left mouse. They also have reduced blood levels of IGF, because if you remember from that previous schematic, growth hormone stimulates the liver to promote IGF production. And so when you can't sense growth hormone, IGF blood levels are reduced as well. And in terms of longevity and lifespan, the GHRKO knockouts have a very much extended lifespan, and that's seen down here in this graph. So in both males and females, if we look at the mean or average lifespan, you can see an increase in the knockout mice that lack growth hormone receptor in both males and females. They live longer. And that's likely due to their smaller body size and probably some smaller metabolic needs, as well as decreased insulin signaling as a result of that growth hormone not being detected, IGF not being able to stimulate insulin pathway, and then kind of extending lifespan as a result. But what's important to note is that these GHRKO knockouts are not the only type of small or dwarf mice that have extended lifespans. There are other dwarf strains, particularly the Ames dwarf and the Snell dwarf, both of which also have a smaller size, have reduced blood levels of IGF, and also have extended lifespans. Additionally, and interestingly, all of these dwarf strains will also have been shown to have a slower rate of aging. So some of their proteins will take longer to denature and deform and become dysfunctional as compared to wild-type mice. The mice themselves have less decline in their functionality. They're able to move better. They retain their memory for longer. They're biologically, quote-unquote, younger than wild-type mice. And they also have a lower incidence of age-related diseases, like cancer. And it's possible that there is a relationship between this growth hormone signaling and the extension of lifespan, as well as the lower incidences of these certain diseases. And you can imagine that, at least from a cancer perspective, growth factors are hormones or proteins that promote growth and proliferation and cell division. And so the inability to sense them effectively, like in a GHRKO mouse, might actually work in favor of cancer, of like slowing cancer development, because it will make your cells less responsive to growth factors and therefore less able to grow and divide. And as we know, cancer is sort of this uncontrolled growth and division of cells. And so it's possible that having less growth factors circulating in your blood, as well as less of an ability to respond to growth hormone signaling, is actually what accounts for that lower incidence of cancer in mice that have the growth hormone receptor knocked out.